Hello and welcome to the cloning revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. Cloning is the production of genetically identical copies of a cell or organism. It is a type of asexual reproduction, meaning no fertilization is involved. Plant cloning. There are two ways that we can clone plants, through cuttings or through tissue culture. With cuttings, a section of a plant is removed with a knife, e.g. a stem or a leaf. The base of the stem of this piece of plant tissue is placed into rooting powder, otherwise known as auxin, to develop roots. And the piece of plant tissue that was previously dipped into rooting powder is now planted into compost, roots will grow and a new plant will form. For tissue culture, a small piece of plant tissue is taken from the plant using a scalpel. These pieces of tissue are known as explant because they came from the plant. The pieces of plant tissue are placed into dilute bleach to sterilize them, then grown on sterile agar containing nutrients and hormones, which result in a large mass of plant tissue called a callus being formed. Different ratios of plant hormones are used to make the callus cells differentiate into roots and shoots, forming plantlets. In the final stage, the plantlets are potted into compost to grow into plants, and each of these plants will be genetically identical, so they will be clones. Animal cloning methods. There are two methods to clone animals: embryo transplants and adult cell cloning. Embryo transplants. FSH, follicle stimulating hormone, is given to the cow by injection to stimulate the ovaries to release multiple eggs. Fertilization will occur where the sperm will fertilize the eggs in the oviduct. After fertilization of each of the eggs in the oviduct, each fertilized egg will divide to form an embryo, and these embryos will form inside the uterus of the cow. The embryos are then collected from the mother's uterus and transferred to a petri dish in the lab. Cells from an early embryo are split before they can specialize. Each cell can then develop into a separate embryo. This embryo is then split, and the cells will divide by mitosis to reform new embryos. These embryos are then implanted into a surrogate mother, and the calves are then born. In stage 4, these calves are genetically identical to each other, so they are clones of each other, because they all came from the original embryo. Adult cell cloning. An egg cell is taken from a sheep. The nucleus is removed from the egg cell, leaving an empty egg cell which is retained, and the nucleus is discarded. A body cell is taken from another sheep, in this case a brown sheep. The nucleus is extracted from this body cell and retained. The empty body cell is discarded. The empty egg cell is fused with the nucleus to form a fertilized egg cell. This fertilized egg cell is given an electric shock to make it divide by mitosis to form an embryo, which is a ball of cells. The embryo is implanted into a surrogate mother sheep. A lamb is born that is genetically identical to the sheep which provided the nucleus, in this case the brown sheep. Benefits of cloning. Agriculture. If an animal has been genetically engineered or selectively bred to have favorable characteristics such as high milk yield, then this animal can be cloned to produce many of them with this characteristic. Medicine. Stem cells can be cloned to produce more stem cells for treating diseases and for research, and this could help replace damaged tissues, organs by cloning cells. Risks of cloning. Reduced genetic variation as all the individuals are genetically identical. This means the populations are more vulnerable to disease. An example is banana trees that are clones. These are currently being affected by a fungus, and cloned animals may have more health problems than a normal animal. Ethical issues. Some people object to cloning because it may cause suffering to animals. It is seen as unnatural, and there are concerns about cloning humans. Thank you for watching. See our revision notes, the link is in the description.